My name is Olivia Cresswell, and I am an undergraduate student in Dr. Kaplan's lab at the Mississippi State Vet School. Dr. Kaplan's lab studies the effects of drugs and chemicals on the immune system. In this poster, I will be discussing the development of an in vitro model to study cannabinoid effects. Important background on this topic includes myelin oligodendrocyte glycoprotein, or MOG. This is a protein that is part of myelin that surrounds neurons. Injecting a MOG peptide with an immune stimulant induces experimental autoimmune encephalomyelitis, or EAE, which is similar to multiple sclerosis, as well as cytokine production in vivo. Marijuana compounds such as CBD and THC have been shown to suppress cytokine production in clinical disease. The significance of these studies is understanding how MOG stimulation affects splenocyte populations and MOG peptide-stimulated cytokine production in vitro, and developing a model in cell culture. We will show that MOG treatment resulted in T-cell increases and B-cell and macrophage decreases sometime between day 4 and 7, and that a subpopulation of T-cells recognized MOG peptide. We present data from 3 experiments. The first examined cytokine production influenced by MOG, CBD, and THC. This then led us to conduct 2 flow cytometry experiments. In all experiments, the approach was the same. We treated otherwise unstimulated mouse splenocytes with MOG peptide 2 times over a 1-week period to see if we could induce cytokine production and observe cell population changes in the cells. This MOG peptide is the same peptide that can be used to induce the multiple sclerosis-like EAE disease. This model is trying to mimic T-cell activation in response to the MOG peptide in vitro. The figure in the bottom left corner shows how different antibodies are used to detect various splenocyte populations, including tetramer-positive T-cells that can recognize MOG-specific T-cells. For our results, Dr. Kaplan initially showed that MOG peptide stimulation of mouse splenocytes 2 times in a 1-week period increased cytokine production, as shown in the interferon gamma results in Figure 1. As you can see, treating with the marijuana chemicals cannabidiol or delta-9 tetrahydrocannabinol at various times over the week can inhibit the MOG-stimulated interferon gamma production. Since we saw that MOG could induce cytokine production, we wanted to investigate which cell population might be responsible. So we used flow cytometry and we tested the hypothesis that interferon gamma produced by MOG peptide treatment of splenocytes comes from T cells. Now on to our first experiment in Figure 2. In each set of flow data, we provide tetramer-negative analysis across the top and tetramer-positive data across the bottom. The use of tetramer-negative versus tetramer-positive did not significantly change the analysis of the other cell populations. In Experiment 1, we noticed that there was about equal CD4, CD8, and CD19 cells at 20 to 30% each. This is different from a normal mouse spleen, where we expect about 10 to 20% T cells and 50% B cells. So this showed that T cells were becoming enriched. Focusing on the tetramer, we saw a slight increase in positive over negative, which suggests that some of the T cells are specific for MOG peptide. In Experiment 2, we analyzed both day 4 and 7. For these samples, day 4 cells only got MOG on day 0, but day 7 cells got MOG on day 0 and 4. The day 4 cells look similar to a normal spleen, but again, day 7 showed the enrichment of T cells and decreased B cells. We also saw a decrease in macrophages over the 7-day period. In summary, we showed that the interferon gamma production seen in the ELISA is likely coming from an enriched T-cell population. Our next step will be to introduce the CBD and THC and see how the cell populations are being affected.